Hello, everyone. My name is ManolisKellis. I'm a professor of computer science and computational biology at MIT and at the Broad Institute of MIT and Harvard. The goal of my research is to understand human evolution and to reverse the circuitry of human disease so we can put an end to evolution as we know it. So I'm a friend of Jay from MIT, a fellow alum, and I'm delighted to speak today about how these circuits and the blend of AI and genomic medicine can enable synthetic biology and rewiring of genomes for the future. In a word, what our lab is trying to do is enable AI for genomic medicine. We're seeking to understand the circuitry of human disease so that we can reverse it. We seek to study the products of evolution so we can help bring an end to evolution as we know it. Evolution is extraordinary, and we can marvel at the products of evolution. We can marvel at the extraordinary body plan of humans, at this extraordinary circuitry of our brain, language, memory, reasoning, and integration, the huge diversity of senses, both for humans and for the natural world, and also more broadly at the early stages of photosynthesis and cellular organization, and this evolutionary arm race between our immune system and pathogens around us. We can also marvel at the simplicity of evolution, the random mutations that generate diversity, and the brutal, cold-hearted selection that leads to survival of the fittest. This brutality can lead to extinction, replacement, winners and losers, and there's a cost to selfishness. A species that becomes too good at exploiting all the resources in its environment can then lead to disrupted food chains and lead to entire ecosystems going extinct. The downside of evolution is that it's also very slow. In addition to this brutality, it just takes billions of years. It's messy. There's terrible genome wiring that results from tinkering rather than synthesizing through systems biology and synthetic biology. Evolution is also brutal, leads to suffering and loss of life, and it's also very inefficient. It throws away entire organisms and entire genomes rather than keeping the parts that are extremely helpful. So this ability to go back and look at the extraordinary machines that have been invented without letting them be lost through extinction. And deep down, evolution is stochastic. It's just a random search. So perhaps with engineering, we can do better. And humans have already been undergoing a different form of evolution. Traditional Darwinian evolution leads to things being inherited vertically. And this has been the case also for humans. In families, in communities, traditionally, these have been in silos. So not only genes have been evolving vertically, but also knowledge has been evolving from grandparents to parents to children, all within the same village, the same family, for the vast majority of human evolution. Resources have also been simply inherited. The house that your grandparents built is the house that your grandkids will grow up with through the vast majority of human evolution. It all has been passed vertically from parents to offspring. What's unique about humans, however, is culture has been traveling horizontally for thousands of years. With the advent of the sharing of stories and eventually writing, the advent of the printed press and Wikipedia, knowledge has been passed horizontally. An invention made in Cambridge, Massachusetts, can immediately travel to Shanghai, China, and from there, been built on and travel to Europe and to Latin America and to Africa, across the world in days and sometimes seconds. Wealth has also been traveling horizontally in humans with taxation and with fellowships. The ability of some parents without children to provide fellowships to extremely talented children from across the world to study somewhere else across the world. And genetics has also been traveling with the ability to move from one city to the other, with the ability to be drawn to an environment with like-minded spirits, and then intermix not only your ideas, but also your genetics through the next generation. Competition in humans has been replaced by collaboration. The diversity of skill sets has enabled specialization, and that allows sharing across broad cultural upgrades, the software, if you wish, to our cognitive hardware. And the result has been this great acceleration from fire, the development of tools, language, writing, ancient Greece and ancient China, the printing, and ultimately telescope, engine, the light bulb, telephone, flight, vaccines, landing on the moon, developing Windows, interactive systems, web, Google, Wikipedia, Facebook, the sequencing of our genome, the invention of YouTube and the sharing of knowledge in milliseconds, self-driving cars, ultimately general AI and being able to land on Mars and other planets. And humans have been using these tools to alter the course of evolution for our environment. We've been turning animals that would eat us to those that can't wait for us to go home. We've turned inedible plants like teosinte into corn, the major source of nutrition in the New World, and selectively bred animals for their wool and for their resources that they give us. But we haven't understood the mechanism through which this human selection of animals works until the last 150 years with Gregor Mendel, who painstakingly worked out the rules of inheritance to understand the lack of blending, to understand dominant and recessive alleles and the independent assortment of alleles in the concept of what we now call genes, the discrete units of inheritance. And that has led to this extraordinary explosion of being able to map the molecular basis of different disorders in very high resolution. But this Mendelian view that has allowed us to find the gene for X and Y and Z is much more complex when you look at complex traits like Alzheimer's disease and cancer and diabetes and obesity and cardiac disorders. We don't have a single gene that governs these. We have thousands of genes whose additive effects can lead to seemingly continuous distributions with thousands of knobs that can be turned. And this extraordinary advance in genetics has led to the human genome, the building of variation maps, understanding of co-inheritance in haplotypes, and ultimately the enabling of genome-wide association studies that have now led to 120,000 dots on the human genome, where we know specific genetic contributors to diversity of human traits. That allows us to now start understanding the molecular basis of these through work in my lab to map the molecular consequences of these genetic differences at the gene expression level, at the epigenomic level, in healthy and in disease samples, to give rise to the ability to recognize the driver genes, regulatory regions, regulators, and cell types that are responsible for these genetic alterations. So we can take an engineering approach to evolution, alter these evolutionary circuits with the new synthetic biology engineering tools. And we are testing these in cell cultures and mouse models to show how this reversal works. To give you one example, we took the strongest genetic association with obesity in the FTO locus and uncovered the driver single nucleotide polymorphism, or SNP, which disrupts a motif that prevents the binding of the ARID5B repressor on a super enhancer that is 12,000 letters long, which then leads to de-repression of IRX3 and IRX5. These, we showed, are master controllers of a process known as thermogenesis that allows your calories to be burned by your fat cells instead of being stored in white adipocytes as lipids. And what we showed is that you can manipulate the circuit to shift back and forth between obese and lean phenotypes like a switch. We can increase or decrease the upstream regulator, leading to a switch between lean and obese. Increase or decrease the expression of the downstream target genes, IRX3 and IRX5, again, switching back and forth between lean and obese. And we can even go and edit a single letter out of 3.2 billion letters in the human genome, and again, switch back and forth between lean and obese phenotypes. This has allowed us to edit human cells from primary adipocytes of risk individuals that have the C allele with a disrupted motif. And by restoring the motif, restoring this T, we have been able to restore thermogenesis and increase it by a factor of 7. We have also shown that decreasing the expression of IRX3, one of the 2 target genes in mice, leads to leaner mice to start with, and mice that are unable to gain weight on a high-fat diet. By understanding the circuitry, we can reverse the circuitry and we can reverse human disease. What that has allowed us to do is now go into many different loci and repeat this process. In the case of oligodendrocytes in Alzheimer's disease, we looked at the strongest genetic association with Alzheimer's in the ApoE locus, and we were able to show that the ApoE4 carriers that have a 10-fold increased risk for Alzheimer's disease are in fact showing impaired cholesterol biosynthesis and metabolism, where the cholesterol accumulates in the endoplasmic reticulum instead of being channeled to the tips of the oligodendrocyte cells to form myelin that protects the neurons. And you can see here the impaired myelination in neurons for ApoE4 individuals. And by understanding that circuitry, we can reverse it with synthetic biology. We can reverse cholesterol transport. We can restore myelination and restore cognition, both in human cells and in mice. We can also use these to understand the circuitry of cancer, to understand how responders to immunotherapy are different from those that do not respond to immunotherapy, and predict a single regulator that we can use in a combination treatment to abolish the recurrence of cancer in mice treated with immunotherapy. We have used these approaches to understand the beneficiary impact of exercise and diet interventions in both human and mouse, to try to recapitulate the effects that we know are affecting multiple tissues and understand what are the cells and the pathways and the genes that are mediating these effects. We've gone into cancer and Alzheimer's and other disorders to understand what are the building blocks of these complex diseases. Can we break them up into smaller parts? Can we understand the transcriptional hallmarks, the individual molecular pathways that are underlying the diversity of Alzheimer's disease? And then use these to discover different subclasses of Alzheimer's individuals, which we can then treat with different combinations of therapeutics. We have developed these modular and programmable CRISPR-based constructs for both editing and for modulating regulatory control regions and genes associated with those circuits, and for changing dozens of these letters simultaneously in the brains of animals. We are now working with Brad Ventuluta and Marinka Zitnik to develop a new language that allows us to seamlessly translate between genomics, machine learning, and chemistry, to understand big language models and geometric deep learning with self-supervised learning, to build these foundation models that allow us to understand the language of protein folding and the language of chemistry, to design next generation therapeutics across many different modalities that allow us to now systematically perturb these circuits. So disease still reigns. We need to dramatically transform pharma. We need to enable personalized medicine by understanding the causal pathways and how these are combined in every individual to enable precision medicine, personalized medicine, and individualized treatments for every patient based on the combination of alterations that they have. And this requires a call to action, a coalition across biology, chemistry, biotech, computer science and engineering, finance, pharma, and bring together patients and hospitals to understand what is the greatest challenge of our generation. And AI can be a partner in this synthetic biology battle to change our evolution. at the genome level to understand the language of DNA, the language of evolution of the epigenome, of non-coding RNAs and single-cell biology, and to reverse and rewire disease circuitry. AI can help us develop new therapeutics through next-generation large language models and graph neural networks that enable us to design drugs that were previously inconceivable through human creativity alone. AI can also help understand the human brain circuitry and become the masters of our brain and augment the human intellectual capabilities through both external and internal means by rewiring in a targeted fashion all of the components that we know influence brain function. This AI can also help dramatically alter education and training and reskilling across teams, across schools, and in a changing job market. AI can also be the personal assistant and coach and partner and ally for each of us, basically design our own goals and become Iron Man through the use of these augmented capabilities to be able to interact with an assistant that never sleeps, that never gets tired. AI can also help us understand and map human knowledge. This is an embedding that we have created of 155,000 papers that have cited our work, clustering them based on their content, based on their projection in this embedding space of AI. This is clustering of 10,000 meetings that we have had, that we have recorded and transcribed automatically through AI, enabling us to now start moving the space of ideas in a seamless fashion. So I am working at this extraordinary institution that is MIT with these amazing computational and experimental science together with extraordinary collaborations with very generous funding. And I'm hoping that the work that we're doing can really help enable the end of evolution as we know it, that we can help enable this ability to generate new understanding of these circuits of human biology and to understand how we can reverse these circuits to enable a new phase of human biology where aging is optional, where disease is a thing of the past. Thank you.